Yes, so mutation order is an important feature of cancer development. And in leukemia, we have the ability to really look at that in a very careful way, particularly with single-cell genomic studies. Many people will be familiar with now single-cell methods for determining somatic variants and somatic mutations in cancer cells. And in the field of AML, there have been 3 or 4 different studies that have been published using these types of technologies to interrogate a number of different patients. What we did was we collated the data from about 130 different patients at several different institutions and started looking at the patterns of mutation acquisition that were determined from single-cell sequencing. And we found that there are canonical patterns of mutation acquisition, many of which could be gleaned from bulk sequencing, but others of which you really don't get until you drill down into the single-cell level, because particularly for mutations that happen at subclonal frequency, you don't know which individual cells they are co-occurring with other mutations. So we found, for example, that epigenetic gene mutations typically occur first, and proliferative pathway mutations like RAS or tyrosine kinase pathway happen later. But that's not always the case. And there are some non-canonical patients in which the proliferative mutations do happen first compared to the epigenetic mutations. Now, our cohort was a little too small to be able to identify clinical features that were different between those patients, but it does raise the possibility that there might be both chemotherapy responses, long-term outcome response differences, and of course, cell of origin and disease pathogenesis differences. So some of our study, I think, is led to new findings that there are these non-canonical pathways, but opens the door to future research for, okay, what does that really mean for patient responses?